Many of you know me. I've been around in the industry for many years, consider myself one of the dinosaurs of EM that's left out there in the field, still working. Been with many places, many companies. I went through a basic, one of the 3 EM schools to start. As we know, we only have Delta College left, but this is where, you know, you really got to learn the field really well, and to progress on to bigger and better things. I went to work at the Yale School of Medicine doing both research and clinical electron microscopy, which of course, both of those involved lots and lots of processing of samples for routine TEM. I've seen a lot of instrumentation come and go over the years. You know, Baltec had one that pumped resins around. That was a failure because, you know, pumping resins and chemicals around through silicon tubes just really didn't work very well. Many of you folks that have been around for a while remember the LKB processor many years ago. That was your typical dip and dunk in a basket. Right now, there's a number of different systems that have come around, but finally, we started working with Heartland Biotech, and this is tissue processing evolved compared to where we were. In most electron microscopy work, we don't like to change anything because it works. In certain cases, changes is not necessarily necessary, but in your tight schedules in your laboratory, consuming large quantity of expensive material to process samples, technician time, evolution is really sort of required to not only improve your results, but to improve your throughput through the laboratory. So a few years ago, I came across working with Heartland Biotech, the Prepmaster 5100, which is an honest-to-goodness robot platform, which improves image quality, convenience, and it's also cost-effective, which is a big ticket item in everybody's laboratory. So most laboratory technicians would prefer to get out of the dull, repetitious processing. I know I certainly didn't enjoy processing 50, 100 samples of brain tissues a day when we did experimental stuff at Yale. In the clinical lab, we would have 10 to 15 to 20 renal biopsies a day. We'd have exposure to all the toxic chemicals that we've been talking about. They're certainly noxious and hazardous, and they're now much more difficult to get rid of. So automation eliminates errors and inherent problems in the manual processing. On the worst day, this machine will beat your best day that you may have in the lab processing tissues, and it'll do it consistently in a routine method. So the solution is the Prepmaster 5100. It's an all-in-one processing system that prepares biological specimens for TEM, volume SEM, serial block phase, FibSem, array tomography, immunolabeling, and SEM processing and dehydration. We usually work with the normal size 1 by 1 millimeter cubed tissue, but we can go up to 1 by 3. We can work with seeded cells on cover slips, organoids, and we can actually even stain grids in the system. The features are, it's a UV block, ventilated, removable enclosure. There's heating, cooling, shaking station. Everything runs off of a laptop with remote monitoring and operation. It has a P2008 channel pipette system that's held on the XYZ gantry of the robot. It processes as little as 8 specimens at a time or even less, up to 96 specimens in 2 hours time. It has now a heated and shake agitation. You can configure the deck however you like it, and it has future upgradability. Folks at Heartland Biotech and I have been discussing some future processes we would like to adapt to this system. So the benefits you can rely on are the quality images, convenience, and effectiveness. This is consistent preparation because you're not being interrupted, you're not pipetting and removing material from vials or whatever the vessel is your sample is in. There's no sample to sample or batch to batch variability. We eliminate human error because everything is in its own little vial. This is especially important in clinical occasions where it's mixing up of biopsies, because the dip and dunk systems, even with the smallest baskets, will still allow a needle biopsy to exit the basket and get lost in the solution or even get transferred to a different basket. This all improves precision and accuracy of results. It improves security, and the system is optimized for cube-sized specimens. Has a very convenient workflow, easy to set up in about 5 minutes, easy to clean up. And as a company that sells a dip and dunk system, the Lynx processor, this is a problem for some of my customers. They don't really particularly either have or desire to take the 15 or 20 minutes to clean all the resin off the different portions of the system. And that leads to errors and problems down the road. With this system, it's very, very easy to clean up, and pretty much everything stays where it belongs. It's completely customizable across the different applications, has a very easy, intuitive interface for protocol development, requires minimal training. It's a very, very simple system to work with. Sample tracking is very easy, as I said. No baskets to assemble or disassemble and track. Each specimen is kept in a discrete microwell for the entire workflow process. Zero crosstalk between specimens, virtually impossible to mix samples up. When we say virtually impossible, but humans load it, so humans make mistakes. Generally speaking, robots don't. So there's still a little bit of human error involved there. Its workflow makes your lab much more productive. You load the reagents in the different wells that are on the deck. You load your samples. You select your protocol and press start, and you basically walk away and allow the machine to do the cumbersome process of processing your samples through into resin, whereas you're off doing something more productive with your time rather than sitting there exchanging media into different vials at different times. We use hydraulic dispensing and aspiration. This accelerates processing without compromising results. As I said, you just press start and walk away. or you can monitor the system from home if you want. It's very safe and reliable to run overnight for greater efficiency. You can monitor it from home if you wish. Prepares 8 1 millimeter cube kidney samples in about an hour into resin. Prepares 96 kidney specimens in less than 2 hours. So you can vary the number of samples that you're running on a daily basis if you're in a clinical lab or in a core facility lab. It's easy operation. You can see your typical tube that we use, and you can see it next to a 3 millimeter grid. The robot itself fits nicely in most fume hoods. There's your sample next to a grid in the shaker agitator, which allows mixing of the sample and the reagent. You can vary the shaking rate and the duration. Everything in the protocol is completely customizable. Okay, so nowadays, you know, you've got safety people hanging all over you, and improving your organizational safety by using this type of robotic system will make your safety people happy. Ventilates into a fume hood. If you can't fit it into your fume hood, it comes with a fume extraction system, which will exhaust your material in your fume hood. You load your toxic reagents at the beginning and walk away. Press the start button. You have pre-dispensed material in sealed reagent reservoirs, eliminating almost virtually your chance of exposure. Also, it minimizes evaporation of volatile chemicals like acetone. Uses standard labware. Nothing's proprietary, expensive, or custom consumables required. You can serve reagent use, save purchase, shipping, disposal costs, unattended operation, frees your time to work on other things more productive in your laboratory. Here's your average cost per specimen, 16 cents. The specimen plates are about $3. Pipette tips, about $10. Water, of course, and ethanol, if you're using ethanol. For 96 specimens, $16. So about 16 cents per sample, which is quite inexpensive. Typical manual preparation time for manual processing renal biopsies. I know this from experience. We used to spend the first half of our day in the laboratory manually processing even as few as 5 or 6 kidney biopsies. We get in at 8 a.m., get the processing started, and then basically have everything in the first change EPON about lunchtime. So the Prepmaster takes 5 minutes or so to load your stations, another minute or two to move your samples around. So it's a 75% decrease in total time that you have to attend to the system, and a 97% decrease in hands-on effort. It'll make your own dilutions of your organic solvents and ethanol, if necessary, right from the stock solutions on the tray. So you're getting the freshest, most accurate dilution right from the get-go. It will use the spent ethanol to deactivate the osmium waste. So this is another major advantage of the Prepmaster. Error prevention. Labs report up to 50% processing failure due to loss or mishandling. Conservative estimate is a 10% loss. Many labs process 2 or 3 specimens to ensure that they get at least a good one. Prepmaster eliminates pipetting errors, never selects the wrong reagent bottle, so a decrease in pipetting error and reagent mishandling to 0, decreases the error rate from 0 from 50%. Some cases, minimum of 10% in most cases. Yeah, many times I've been at the hood working, you get interrupted, you get a phone call, your sample sits in a reagent for a longer period of time, you grab something inadvertently, next thing you know, you have a messed up process. Benefits you can rely on, it's automated, reproducible, standardization, it's easy to set up, it's easy to clean up, it's very efficient with your time, it's safe, your safety people will love it because it minimizes exposure, it's cost effective, uses much less reagent than you would in your traditional dip and dunk system. I know the Lynx processor that we also have available, you need at least 18 ml to process, 10 to 12 samples, which is quite a lot of reagent. Not picking on any one particular system, any of the dip and dunks rely on an automated system that moves things around. If you don't keep them clean and free of sticky resin, they break down often. They have limited capabilities. Again, they're unevolved. I mean, this type of system goes back many, many years, whereas the Prepmaster is a specimen processing evolved. Very wasteful, uses more reagent, complicated to set up, messy to clean up. Reagents are much higher cost because you're using more, and because you're using more, your disposal cost is much higher. Prepmaster uses about 3.5 ml of reagents per run, where your automated processor of dip and dunk type use 30 ml, which is a 90% reduction in use of reagents. So this is just a comparison between the Prepmaster and your traditional dip and dunk automated system that's available, and even EMS has one available. Takes several hours to run renal biopsies, whereas Prepmaster, 8 samples in less than an hour, double that time for up to 96, 0 failures. So far, the Prepmaster has been out and tested for many years now. There's been no breakdowns at all in the systems that have been fielded. I have to admit, even our Lynx has issues, and quite commonly, but usually 99% of them are related to on how clean the user keeps the system. The resins have a tendency to cause the greatest mishap rate in the system. Again, the reagent use is 3.5 mils per run versus 30 mils per run. Reagent cost is 90% less. Again, we have a modern system and not an antiquated design that's been around for 20 or 30 years. Setup is easy, whereas the dip and dunks, you have to program on a little keypad. We program ours on a laptop that comes with a whole bunch of recipes already loaded. All you have to do is just load it and basically hit run, or you can create and modify your own protocol to your liking quite easily. I figured out how to use it in a very short amount of time. It was very intuitive and pretty slick interface. Hazardous waste disposal is a lot less. And like I said, you're working on a Windows laptop computer, which comes with the system with everything all preloaded and set up versus some keypad either on the machine or something that you can hold. Again, you can rely on image quality, convenience, and effectiveness with the Prepmaster. The results are consistent, pristine ultrastructure. The results say it all. Some images will follow from mouse ovaries, taproot plant, hippocampus, cardiac muscle, astrocyte grown on cover slips, liver biopsies, kidney biopsies. Here's some results. There's a number of published publications that are out involving the system. Testimonials from some of the people using it. You can run experiments with up to 4 variables in one simple computer-controlled, precise, automated experiment. In this way, you can properly compare results from those 4 different variables. We can't do that by hand. Here's some publications that you can look up. And I believe this year there's going to be another couple of presentations, posters at M&M, if I'm not mistaken. These are astrocytes on cover slips. If anybody's ever processed cover slips, you know how cumbersome it is pulling out the cover slip out of the well. Well, when you can process them without any error, without removing the cover slip, getting perfect results every single time with 4 different variables, this is a significant evolution of processing EM samples. Biopsies and cell pellets are easy to do. If you don't want to embed them right in the wells that you process them in, which you certainly can, you can easily remove them and put them in flat embedding molds, however you like to work with them. If you need orientation, like for this nasal cilia biopsy, which I've had experience doing myself many years ago, was always fun trying to orient cilia so the pathologist could look for the syndrome, which causes defective cilia in the nasal passages of young children, not clearing their sinus passages, causing lots of sinus infections in the poor little kids. Okay, a little fast here. Beautiful skeletal muscle. Done at the Medical College of Wisconsin, M-line, A-bands, I-bands, all the sarcoplasmic reticulum, renal glomerulus, red blood cell, basement membrane of the podocyte, some reactive processes over there. We won't get into the diagnostics, but this is all done routinely and quite cleanly in a very short amount of time. Again, nasal scrapings from infants with that ciliary disease that unfortunately is genetic. I don't know if they've figured out a genetic way of reversing that or not, but these are from cell pellets, the Medical College of Wisconsin. I'll let you read through this. They're doing both diagnostic and research work. It's streamlined their preparation and put the technicians back doing things that are more productive. It saves time, consistent results, cost-effective, enhanced reproducibility, all those things that are desirable for your facility. What next? Well, we have, for those people who want to still embed in beam capsules, we're developing a beam capsule holder that you can use. This way you are working with a more familiar platform. Works the same way as it does with our plates. Same great results, same savings in reagents. It's just that some people are hung up on beam capsules. Other people are open to using other types of embedding molds. I believe also for the Mayo Clinic, they wanted to stick with dip and dunk, so they're using traditional baskets on the Prepmaster, and they're moving it from vial to vial. So very flexible platform. We're now working with the MPAR database. We're starting to get integrated with that, so you can upload your protocols and you can download protocols from the central database. It's quite useful to have an online resource where you can get and send protocols that you may develop. Sharing with others is really, really important in this field. We're sort of operating in isolation. Most of us only really sort of get together for the major meeting, M&M, and this is a good way to collaborate online. Kendar is one of the main people at NCI in Frederick that works with this sample preparation widget. Also Michelle Darrow and Charles Bosch Pignon, all involved in putting together this sample prep working group, which is essential for our industry because there's too many newbies contacting the EM vendors for very simple questions. So this is an important database in moving forward to help everybody produce the best samples they possibly can. So you can basically go down from our platform, you can enter pH, sample temperature, all sorts of important, sometimes overlooked process elements for your protocol. And this can all be uploaded and downloaded and used interchangeably with the 5100 Prepmaster. So here's a database sample. So you can pick amongst the different methodologies. You can input your data for step 1 and on through the entire protocol development process. So summarizing, the 5100 provides reliable, reproducible, effortless, error-free, economical, safe execution of your EM workflow in the sample prep realm of your laboratory. And now we'll entertain some questions if anybody has any.
Can I ask a question?
Sure, of course.
Is there a preferred resin to use for this robot? Do you have to use like low viscosity resin, or you don't think there's any difference, or you have to program different program when you change resin?
You can use pretty much any of the resins in there. I don't know how many different ones have been used off the top of my head. But one of the advantages that is with the infiltration steps, because you don't necessarily, certainly you're not going to use propylene oxide in this. You would use acetone, which is also volatile, but all the little wells on the plates are sealed that the pipette tip pierces and moves reagents around. But we've had great results across the board with different resins. I don't know if we've worked with any of the acrylics in it yet, or it certainly would work with any of the epoxy resins. That's what most of the work so far has been done with.
Okay, all right. There's someone on the chat asking about the cells in pellet. You want to answer, or Tom, you want to answer, or you have already
Actually,
answered?
Rob would probably be the best if you don't mind. Rob Goodwin, would you answer that question?
Yeah, so two things with the resin really quick. We just use a regular EPON mix, but we have switched to the hard resin plus kit that you get from EMS because it's just a little more fluid. So the way we process it is we just do a 50-50 of acetyl nitrile and resin, and then we do 2 changes of 100%. So the 100% you do have to remove by just a little pipette tip. The hard resin plus kit just makes it a little bit easier to get out of there. And it's real easy to mix up. You don't have to measure anything. You just dump it all in and mix. Everything's pre-measured, so it saves time there too. As far as the cell pellets, basically all I do is I spin them down into appropriate, I spin them down in my tubes, break them up into appropriate sizes, because that is one thing is if they are too big, you kind of pierce them with the pipette tips. So you do have to keep them in the smaller size, but that's where that beam capsule holder will probably work very well. But what I do is I embed them in histogel first. So that keeps anything from breaking up, and then you can just cut them, embed them in the histogel, cut them into appropriate sizes, and then just process as normal. The histogel allows all the chemicals to pass through, and then you just embed it. And I've had, so those cilia that you saw, those were done in that process in the histogel.
Very nice. I'll talk briefly about acetonitrile since that was brought up previously. I've advocated the use of acetonitrile for a number of years, mostly because of our links processor, because even though there's a cover that comes down and seals the vials, a lot of the volatiles like acetone and certainly propylene oxide, by the time you go through the 4 or 5 hours of the process, most of your propylene oxide or acetone has evaporated. Acetonitrile, you can fill ahead of time, and it will still be there because it's not quite as volatile. Yes, it costs a little bit more money, but it's a lot better than putting your samples in a dry vial at the end of the longer process. And as someone previously noted, there's almost no visible difference between using acetonitrile and your traditional sort of intermediate agents. Matter of fact, we should do a double-blind study and have a poll and see if somebody can guess which is using the regular intermediate agents and acetonitrile. I bet it's very difficult to actually come up with the ones that use the acetonitrile. It's really quite good. It's a little bit more expensive, certainly, than acetone, Propylene oxide, I don't like working with because of volatility and toxicity, as well as its unfriendly nature, being quite explosive. I joke let me know with the staff over in the chem lab when they're filling propylene oxide from the big, let me know so I can be off campus visiting a customer or something, because it's, you know, quite a hazardous chemical to work with. Has very nasty tendencies to make big holes in the ground when those go off.
Thank you, Al, for the clarification. There's some comments in the chat, and if there's no any other question, I think we can close the session. And I want to thank everyone that staying all the way to the end. I apologize for we went over time. And thanks all the speakers, and thanks everyone who attended the meeting. And I hope to see you in 2 months again. And do send me any feedback and to make BioEMTalks even better. Thanks.
Thank you, everyone.